Hi, I present to you histology of the neuron, spinal cord, cerebrum, and cerebellum. I'm Dr. Aidoo with the Department of Anatomy and Cell Biology, School of Medical Sciences, University of Cape Coast. So the neuron or nerve cell is made up of this spherical body you see here. It is the cell body or soma. It contains the nucleus, colored blue here. Is usually dark or black. In this case, it's colored blue. And then the cytoplasm, within which the nucleus is situated. So the cytoplasm is within, and then the nucleus lies inside the cytoplasm. The extensions you find there is known as the dendril. The plural is dendrites. So several of them are the dendrites. They are the input for the neurons, which means that they receive signals or stimuli and pass it on to the cell body, and then it is carried along the axon, which is the relay. In a histological micrograph, you need some special stains to pick up these neurons we just saw in the previous slide. So let's just transpose the understanding of the previous slides, which was just a drawing, to this micrograph. You can see that you have this region which is the cytoplasm with the nucleus. Okay, so the cell body is there. You can also see some extensions coming out of this cell body. So some of them will be dendrites and one of them will be axon. So usually it's difficult to tell which one is the axon or dendrite, but when you look at it critically, you will know that the axon has an axon hillock, which is devoid of nasal granules. So usually that part will be paler, okay, and that will give you an indication of the axon. You can also see mesh-like structure in the matrix. Okay, so you have connective tissue with glial cells, which are supporting cells. Some of the supporting cells are indicated by this arrow. You have another one here. So there are about 3 of them which have been shown, but there are several of them which do not have any arrow pointing at them. The neurons, as I've indicated, you can easily see them by their stellate appearance, star shape, okay, with the cytoplasm and the nucleus. There you can see there's a nucleus within and then the cytoplasm, and these are extensions, so they are dendrites. You can see another one here. So with this particular stain, you can actually identify the axon, okay, and the axon hillock. All these are nasal bodies or granules that are contained within the cytoplasm. That is the nucleus. So that is the axon, and you can see it is paler than the rest. These are dendrites. That's another one there with a D, and there's another one there. Now, you shouldn't mistaken a long dendron to be an axon. Always look at the axon hillock. See whether there is nasal granules or nasal bodies present or absent. So it is named, the nasal bodies or granules are named after Franz Nissel, who discovered it. So with this drawing, you can easily appreciate that. Okay, a paler segment, the axon hillock, which continues as the axon. You can always tell how a neuron or nerve cell looks like in a micrograph. Now, what you see here is not showing dendrites and then cell bodies. It's rather showing the processes of an axon. Okay, now you can see where the arrow is pointing at. There's a constriction and a line in between, showing or indicating the node of Rambia between one myelinated area and then the other. These are nuclei embedded within the tissue. In this very nice stain, the spinal cord, which is also made up of several neurons or nerve cells, and especially the uh, fibers or axons of the nerve cells, will be concentrated within the spinal cord. In the area colored blue is the white matter, and then the area within this H-shaped structure is the gray matter. So the white matter is divided into a lateral funiculus of fasciculus LF, a dorsal funiculus of fasciculus DF, and then a ventral funiculus of fasciculus. So that's the gray matter, that's the central canal. That is the anterior median fascia, and then that's the posterior median sulcus. So usually you have a lot of myelinated axons coming into the spinal cord and filling up this place. That is why it is known as white matter. And then the gray matter is due to accumulation of cell bodies. Okay, it doesn't mean you don't have axons there at all, but the number of cell bodies there outweigh that of the axons. And here too, you have a lot of myelinated axons, but a very few number of cell bodies. So for this staining too, you can tell the white matter and then the gray matter. The gray matter is divided into a ventral and then a dorsal horn. So ventral horn, dorsal horn. When you look at the dorsal funiculus, you can see that there's a portion which is demarcated from the lateral portion by a sulcus. That region you find here, that is the dorsal funiculus, is also called the posterior column. Posterior column. And you usually find the fasciculus cuneatus or the fasciculus gracilis. The fasciculus gracilis is usually found in the entire segment of the spinal cord, but the fasciculus cuneatus is usually found at the upper segment from T6 upwards. You don't find it below T6. And they are actually the additions of sensory fibers at the upper portion of the body being added onto the spinal cord ascending column. How do you identify spinal cord segments at different levels? You use these key points. The amount of white matter is greater in the rostral segment, that is the upper segment, than in the caudal or lower segment of the spinal cord. And the amount of gray matter is greater in the cervical and lumbosacral enlargement than in other segments. Why? Because in the cervical enlargement, you have the brachial plexus. So that one will give you a lot of motor innervations, okay, within the gray matter. A lot of innervation going into all the muscles in the upper limb. And all those fine little lumbricals in the hand all need nerves. So you have a lot of motor supply within that region, just as the lumbosacral region as well. Because of the lower limb, you need a lot of nerves to supply the muscles there, so you have those enlargements in their gray matter. The lateral horn is evident only in the thoracic segment, as we will see indicated. So that is the lateral horn showing on the lateral sides of the gray matter. Now you can look at this from sacral to cervical, and it will be coincident with the sentence we read. The amount of white matter is greater in rostral segment than in the caudal segment. So you can see the white matter here is larger, okay, relatively, than as you move down to the sacral region. Also, the cervical region of the spinal cord is oval in shape, and as you move down to the sacral portion, it becomes rounded in shape. In the lumbar portion, because of the lumbosacral enlargement, you can see that the ventral horn, which carries most of the motor fibers which are going to innervate the lower limb, are present, and hence you have a very large gray matter, especially in the ventral portion relative to the others. You can also see it for the sacral. You have a darker, bigger ventral horn, also because of the lumbosacral enlargement. The thoracic is relatively small in terms of its ventral horn, and the dorsal horn very small. Then for the cervical, because also the brachial plexus and then the motor innervation of the hand requires a lot of motor fibers, you also have this enlargement. So this is a very fine arrangement. You can look at the gray matter in proportion to white matter at the various levels of the spinal cord, from the sacral. Okay, so this will be lower sacral, this will be upper sacral, that close to the lower lumbar, and then you have upper lumbar, then uppermost portion of the lumbar, and then that will be thoracic because of the presence of the lateral horn. You can also see portions of the lateral horn, so these two will be thoracic. Then this is cervical region. Observe the very big ventral horn. So a similar trend follows in this. So this is cervical enlargement with a large ventral horn, and then thoracic small ventral horn but with a lateral horn. Then you have the lumbosacral enlargement. When you look at this portion, the dorsal portion here, okay, or the posterior column, you can see the fasciculus cuneatus and fasciculus gracilis. But in the lower portion of the thoracic, if it's especially below T6, then you are going to have only fasciculus gracilis showing, and obviously in the lumbosacral you obviously have only fasciculus gracilis showing without fasciculus cuneatus. But here you can see fasciculus cuneatus, fasciculus gracilis. So these are representations of C5 to T1, and then L2 to S3. So C1 all the way to T1, T2, and then you have the L2 to S3 also showing. When we look at the cerebral cortex, we find very large neurons which are usually multipolar neurons. So you can see the nucleus, the cytoplasm, and then extensions of the dendrites. Then again, you can see glial cells there. So all I want you to appreciate most is how to identify neurons. That is the white matter and gray matter. That is the internal capsule, thalamus, also situated there. The cerebellum is quite moderate because it has only 3 layers. You have the molecular layer, Purkinje layer, and then granular layer. So the molecular layer is basically made up of the dendrites of the Purkinje cell. Okay, I mentioned Purkinje cell not long ago when I talked about the cerebrum. So these are the dendrites of the cell body of the Purkinje cell. So the Purkinje cell, if you have several of them, it's going to make this place darker. Okay, and that becomes the Purkinje cell layer. And then here you have the granular layer, which is made up of mostly fibers. So when you look at this, you can see that the cell body of the Purkinje cell is located along that line. Okay, and you rather have extensions or fibers mostly in the granular layer. You also find some basket cells. It's also a type of neuron. Okay, you can see that there. In this micrograph, you can easily identify the cell body of a neuron, and that is the Purkinje cell. The granular layer is that region. Then the molecular layer will be containing a lot of the dendrites from the Purkinje cell. The cerebellum contains these layers as we have seen. However, the very nice architecture, the arborization of these, architecture is very, very peculiar to the cerebellum, cerebellar cortex. So the molecular layer, the Purkinje cell layer, and then the granular layer. Here we can see the basket cell. You also have some stellate cells. Parallel fibers are also located in the molecular layer.